When we examine human history through archaeological evidence, we encounter a timeline paradox that challenges conventional evolutionary models. For most of human existence, spanning hundreds of thousands of years, technological development progressed at an extraordinarily slow pace. The archaeological record reveals that early stone tools remained remarkably consistent in their design and function for immense periods of time, showing only minor refinements across hundreds of millennia. The Old tool industry, characterized by simple stone choppers and flakes, emerged around 2.6 million years ago and persisted with minimal change for nearly a million years. The Acheulean tradition, recognizable by its symmetrical hand axes, appeared approximately 1.7 million years ago and remained dominant for over a million years. This consistency suggests the cognitive capabilities of their creators remained relatively stable across countless generations. This pattern of technological stasis presents a significant puzzle. If the human brain evolved gradually, we would expect to see a corresponding improvement in technological sophistication. Yet the archaeological record shows long periods of minimal innovation followed by sudden accelerations that cannot be explained by gradual genetic changes. Imagine a world where technology barely changes for hundreds of thousands of years. Then suddenly, around 50,000 years ago, human innovation accelerates dramatically. This period, known as the Upper Paleolithic Revolution or Creative Explosion, transformed our understanding of cognitive evolution. Tools that have remained virtually unchanged were rapidly replaced by sophisticated implements designed for specific purposes. Blade technologies, bone tools, fishing equipment, and advanced hunting weapons appeared with remarkable suddenness. This acceleration intensified around 30,000 years ago, when the archaeological record shows widespread emergence of fully modern behavior patterns. Figurative art, musical instruments, complex tools, personal ornamentation, and evidence of ritual practices appear across continents with a suddenness that archaeologists struggle to explain through conventional evolutionary models. The cave paintings at Chauvet Cave in France, dated to approximately 32,000 years ago, exemplify this sudden sophistication. These artworks display advanced techniques including perspective, shading, and anatomical accuracy that suggest a fully developed artistic tradition without clear developmental precursors. Archaeologist Jean Klotz noted, "The artists of Chauvet were as technically accomplished as any that followed them. Their work represents a profound and sophisticated aesthetic sensibility that appears fully formed in the archaeological record." Similarly, Venus figurines, small sculptures depicting female forms, appeared across Europe between 35,000 and 25,000 years ago with consistent stylistic elements despite their wide geographical distribution. The bone flutes discovered at Geissenklosterle Cave in Germany, dated to approximately 36,000 years ago, feature evenly spaced fingerholes capable of producing specific musical scales, indicating both technical skill and theoretical understanding of musical intervals. The dramatic turning point in this timeline creates a profound paradox. Anatomically modern humans had already existed for over 150,000 years before this creative explosion occurred. Fossil evidence indicates humans with essentially modern skeletal anatomy appeared in Africa around 200,000 years ago. Yet the archaeological evidence for modern behavioral patterns didn't consistently appear until much later, creating a gap of at least 100,000 years. Paleoanthropologist Ian Tattersall addresses this disconnect. There's a huge disconnect in timing between when we became the anatomical creatures we are today, and when we started behaving in a symbolic manner. That's a gap of at least 100,000 years that's very difficult to explain. This pattern suggests certain cognitive capabilities may have been activated rather than evolved, that the genetic potential for advanced cognition existed long before being fully expressed in human behavior. Modern human cognitive capabilities may have emerged rapidly when specific neural systems reached completion or were triggered by environmental or cultural factors. The evolutionary timing paradox becomes even more striking when examining specific innovations. Sewing needles around 30,000 years ago enabled tailored clothing, significantly improving survival in cold environments. Advanced projectile weapons like the atlatl dramatically increased hunting efficiency. These innovations appeared suddenly rather than through gradual refinement. This acceleration in innovation occurs too rapidly to be explained by genetic changes alone, which typically require thousands of years to spread through populations. The flourishing of technological innovation observed in the archaeological record happens faster than genetic evolution would allow. The paradox raises fundamental questions about human cognitive evolution. If the genetic potential for advanced cognition existed in humans for over 100,000 years before being consistently expressed, what triggered its eventual emergence? Why did human creative and technological development remain relatively static for thousands of generations, then suddenly accelerate within a few millennia? This explosion of human creativity forces us to consider alternative explanations. Was this sudden flourishing the result of a genetic program activating at a specific developmental point? Did cultural factors reach a critical threshold, triggering innovation? Whatever the explanation, the timing paradox in human technological development represents one of the most significant puzzles in our evolutionary history. The plasticity puzzle. The human brain possesses a remarkable ability to reorganize itself throughout life. This capacity for neuroplasticity, the brain's ability to form new connections and adapt to new information, represents one of the most distinctive features of human biology. While many species exhibit some degree of neuroplasticity, humans display this trait at levels that far exceed those of any other organism on Earth, including our closest primate relatives. Neuroplasticity occurs when the brain creates new synaptic connections or modifies existing ones in response to experience, learning, or injury. Most animals experience plasticity primarily during early development, with brain malleability decreasing as they mature. Cats require proper visual input during a critical period to develop normal sight, after which their visual cortex becomes significantly less adaptable. Humans break this pattern completely. Our brains maintain extraordinary plasticity throughout our entire lives. This exceptional flexibility enables us to learn new languages in adulthood, master complex skills decades into our lives, and recover function after brain injuries. Consider stroke patients who relearn speech through therapy as their brains form new neural pathways to bypass damaged areas, a testament to our remarkable neural adaptability. The differences between human and animal plasticity are stark. Humans, lifelong learning capacity with minimal decline. Primates, significant learning ability that diminishes substantially with age. Other mammals, critical periods after which plasticity sharply decreases. Birds, reptiles, limited plasticity primarily confined to developmental windows. This lifelong plasticity appears specifically programmed into our genetic code through regulatory elements that control how our neural architecture develops and adapts over time. The regulatory mechanisms controlling human neural plasticity show unusual characteristics that some researchers suggest resemble engineered systems more than products of random mutation and selection. Dr. Marcus Pembry, professor of genetics at University College London, notes, the genetic mechanisms controlling human neuroplasticity show a level of organization that's difficult to explain through conventional evolutionary processes. These systems maintain developmental stability while enabling extraordinary adaptability. The plasticity-led evolution model proposes that phenotypic plasticity, the ability to produce different physical characteristics in response to environmental conditions, plays a crucial role in evolution by accelerating evolutionary change. North American spadefoot toads illustrate this concept, with tadpoles developing as either omnivores or carnivores depending on their environment, allowing survival in varied conditions. Human neuroplasticity, however, operates at an entirely different level. Our brains contain specific regulatory elements within non-coding DNA that control plasticity across multiple scales, from individual synapses to entire neural networks. This sophisticated control system enables continuous neural reorganization throughout life, creating new pathways in response to experience and learning. These mechanisms allow humans to acquire skills, languages, and knowledge far beyond what other species achieve, despite having similar neural building blocks. Chimpanzees share approximately 98.8% of our DNA yet display dramatically reduced neural plasticity. While they learn impressive skills, their capacity for continuous adaptation remains limited compared to humans. Language acquisition clearly demonstrates this difference. Children naturally acquire whatever language they're exposed to, mastering complex grammatical structures without explicit instruction. Though this ability decreases with age, adults can still learn new languages throughout life. Chimpanzees, despite intensive training, never develop comparable linguistic abilities due to less flexible neural pathways. Human neuroplasticity appears front-loaded in our genome, programmed to activate specific cognitive capabilities at certain developmental stages. These regulatory systems ensure that certain neural networks form during critical periods while allowing others to remain adaptable throughout life. Language networks develop during specific windows in childhood but maintain significant plasticity into adulthood, maximizing learning potential. Research comparing gene expression in human and chimpanzee brains reveals significant differences in genes controlling synaptic plasticity, particularly in the prefrontal cortex. Many of these differences involve human accelerated regions, HARs, DNA stretches that remained largely unchanged in most mammals but evolved rapidly in humans. A disproportionate number of these HARs regulate genes involved in brain development and neural plasticity. This extraordinary neural plasticity raises profound questions about human cognitive evolution. Was the human brain specifically designed to be a learning machine? The genetic programming enabling our continuous cognitive development appears remarkably sophisticated, with regulatory systems maintaining a precise balance between stability and adaptability throughout life. The significance becomes clearer when we consider what this plasticity enables. Humans build cumulative culture, each generation learning from and building upon previous knowledge. This requires specialized neural adaptations supporting acquisition and transmission of complex information across generations. Whether these genetic mechanisms emerged through conventional evolution or other means remains one of the most fascinating questions in human origins research. What's clear is that the regulatory elements controlling human neural plasticity represented defining features separating us from all other species, enabling our unique capacity for continuous learning, the foundation of human culture, technology, and civilization. As we've explored the mysteries within our DNA, we face a profound choice: accept conventional evolutionary explanations despite their significant gaps, or consider the possibility that our genetic code contains evidence of intervention we're only beginning to detect. The anomalies we've examined raise questions that current science struggles to answer completely. Our remarkable cognitive abilities and complex genetic architecture point to a deeper story. Will you continue to marvel at the secrets hidden in your own cells? Perhaps our greatest mystery lies not in distant stars, but within us, written in a genetic language we're still deciphering, with chapters of human development waiting to be understood.